Hi everyone, I'm Thiru, and today I'm going to take us back into the Archean world. We're going to look into silica isotope fractionation during the reductive dissolution of an iron silica gel. And this reduction is mediated by an organism, desulfuromonas, which is capable of ion reduction. And before we proceed any further, I would like to just thank UW-Madison and also NASA for allowing me the opportunity to be a part of this study. All right. Before I jump into any details, I'll just give you a brief outline of my talk today. So first of all, we're going to look into two papers from the literature that served as a motivation towards this iron silica gel study. And then we'll move into the Precambrian silica cycle and contrast it with the modern silica cycle and look at the difference. And then we'll go into some silica isotope basics, looking into silica isotope abundances, delta silica notation. We'll proceed into looking why is it that silica isotope is an important factor in the Precambrian record. Why is there interest for silica isotopes in the Precambrian record? So the first example we would look at is the coupled iron and silica model, and then we'll move into silica isotopes in cherts that have been particularly used as paleothermometers. We'll also go into silica isotopes as a source tracer. We'll wrap that up, and then we'll move into my experimental approach, looking into how I synthesize the iron silica gel and also the artificial Archean seawater, which is the seawater matrix media here. And then I'll show you some data. We'll go into the ion reduction of this gel over time. And then we'll move into isotopic data in the sense of silica isotope data for the aqueous and solid phase of my samples. We'll conclude with some fractionation factor over time and what that means to us, or at least to me at this point. All right, I mentioned earlier that there were two papers from the literature that sort of motivated the study. The first paper looked at iron isotope fractionation of this iron silica gel or coprecipitate in an aqueous media similar to the Archean seawater, and found that the iron silica gel is of highly reducible nature. And the silica present in this gel leads to formation of silica-bearing products, say, for example, precursor to greenolites in banded iron formations that we see today. And then the second paper also looks into iron isotope fractionation, but looks into iron isotope fractionation between the aqueous ferrous iron and also iron silica gel in an aqueous media of seawater composition from the Archean, and found that by varying iron to silica ratio in the gel, you get different fractionation factors. So when you have an iron to silica ratio that is high, you have a lesser negative fractionation factor as compared to when you have an iron to silica ratio which is smaller, you see a more negative fractionation factor. Both of these papers look into iron isotope fractionations though. But what about the silica that's present in this gel? So what is the silica isotope fractionation that you see when silica is mobilized during disseminatory ion reduction? So that's what we're going to try to answer with my research. But before we jump into answering this question, let's look into some background information. Sorry about that. So I mentioned earlier that we're going to look into the Precambrian and also the modern silica cycle. As we've seen from the previous talks, one of the major differences between the Precambrian and modern silica cycle is the presence of biology. So in the Precambrian, there was no biology. There was no silica-secreting organisms present. Therefore, there was a high concentration of silica in ocean waters. So precipitation of amorphous silica in sedimentary layers in basin waters was possible, and that is evidence of the cherts that we see today. Some silica isotope basics. So there are 3 stable silica isotopes, with 28 silica being the most abundant one and 30 silica being least abundant. Delta silica notation is presented in the traditional per-mil notation. Silica isotopes in the Precambrian record. Why is it of any interest? So the first model here is the iron and silica cycle model that was proposed by Fisher and Connell. So they proposed that in the phoric zone where anoxic photosynthesis takes place, you have ferrous iron, which gets oxidized into ferric hydroxides. And then the silica dioxide in ocean waters that has a high affinity to sorb to ferric hydroxides. Once the sorption takes place, the ferric hydroxides then sinks to bottom water and undergoes reductive dissolution. With reductive dissolution, we have a ferrous component of iron, and we have the silica dioxide that can then combine and form iron silicates. Again, we see iron silicates are precursors to minerals that we see in banded iron formations today. Moving on, silica isotopes have also been utilized as paleothermometers from the Precambrian record. For example, Robert and Chalcedon used the correlation between delta-18 oxygen and delta-30 silica to actually trace the temperatures of ocean waters back to the Archean. And the next example of silica isotopes from the Precambrian record would be as a source tracer. As we can see here, that there are two different types of cherts based on their chemical composition. One is the S cherts, and the other is the C cherts. The S cherts is said to be from silica-saturated seawater that has been continuously responsible for silicification. And the second one is chemical cherts, which represents a mixing gradient between the hydrothermal inputs and seawater. We've seen that silica isotopes is affected or controlled by temperature sources. But what about the processes that affect them? What about biology? That's where my experimental approach comes in. So we have the iron silica gel and the artificial Archean seawater representing the seawater composition of the Archean. So for the iron silica gel, I used 3 of the ingredients above, allowed them to mix with open atmosphere for about 2 weeks on a shaker. Once the acid extraction accounts for about 95% of the ferric component to total iron, that is in close representation of the iron tree phases that might have been present in the Archean environment. And then for the artificial Archean seawater, it's just the recipe modified from the first motivational paper that I showed you guys. It's basically made to be anoxic and low in sulfate content, and we did not introduce any silica in this aqueous phase because we didn't want any of the minor signal released by the microbes to be blocked when we analyzed it for silica isotopes. Desulfuromonas is the organism of choice in our experiment, and the reason being so is because Desulfuromonas is the marine cousins of Geobacter, which have been recognized for iron reduction. And previous studies in the literature have proven that they are capable of reducing ferric iron in components which contain silica also. So these are my experimental vial. As you can see from day 1, I have 4 experimental vials. 2 vials contain cells of desulfuromonas, while 2 other vials are just the gel and the artificial Archean seawater. At day 30, it becomes really obvious to us that which vials contain cells and which vials are the control experiments. You can see the coloration difference here. All right, so as I've mentioned earlier, the coloration difference are the first 2 vials. So they transform from being yellow to a little bit of dark greenish. That is the vials with the cells. That shows us that the ferric component in the gel has been reduced to the ferrous component. And in the control experiment, since there were no cells of desulfuromonas, the coloration remained the same, and there was no presence of ferrous iron, or there might have been very insignificant amounts of ferrous iron from the initial iron silica gel. This is the iron reduction data for the iron silica gel over time. And as you can see, corresponding with the coloration from the first 2 vessels, cell 1 and cell 2, you can see that the reduction increases in that cells. The reduction is at about 60 to 70% over a period of 70 days. As for the control experiment, the ferrous iron presence is insignificant. It's about 2 to 3%, and it remains stable. It doesn't fluctuate much. So that shows us that reduction hasn't taken place in that 2 control vials. All right, this is the silica isotope data for the aqueous phase and the solid phase for my samples. The general takeaway from these 2 plots would be, for the aqueous phase, you see an enrichment in delta-30 silica, but for the solids, you see a depletion in delta-30 silica. But the other thing that you want to take away from these plots is when you look at the aqueous phase, you can see that the enrichment for delta-30 silica for the cell experiment is slightly higher than that of the control experiment. And that tells you that presence of ferrous iron is causing that difference. With the aqueous and solid data for the silica isotopes, we can actually calculate the fractionation factor over time. And these are the numbers that I've came up with. So I'd like to point out that for the cell experiment, you see a larger fractionation factor as compared to the control experiment. And again, that is due to the presence of ferrous ion in the cell experiment and the absence of ferrous ion in the control experiment. Therefore, we can conclude that ferrous ion, when it's produced by reductive dissolution of this iron silica gel, causes a larger fractionation factor as compared to a vessel with no ferrous ion or minimal ferrous ion. And the isotopic fractionation here is probably not controlled by biology, but the magnitude of this fractionation is more attributed towards the absence or presence of ferrous ion. Therefore, it's not only temperature or sources that's an influence towards silica isotopes, but it's also the process that mediates this. So that's pretty much all I have. Thank you.
Okie dokie, we have time for a few quick questions. Do you have any idea how easy it would be to find evidence for this sort of increased fractionation in the actual fossil record?
In the actual fossil record?
Yeah, looking at actual iron-banded formations, would this be something that would survive that long?
I know Andy Chaya, this person, has looked into fossil records in the Precambrian, but I don't necessarily know if he has looked into silica isotope fractionations. So, yeah.